Hello again. Today we continue with a particular topic about immune system. We're going to discuss today adaptive immunity. So in the previous chapter we discussed innate immunity, and this picture right here is giving you kind of like a big picture of how innate and adaptive immunity work over time. Remember, I mentioned during the first class that it is impossible to separate these two important components of the immune system. When we get exposed to a microbe, let's say here, and let's say the microbe is able to cross the first line of defense of the innate immunity, the epithelial barrier. And at this point, the cells that have pattern recognition receptors, they will sense the presence of that microbe, and they will start to produce signals that will contribute to start the effector functions of the innate immunity, but also start the communication with the components of the adaptive immunity. These cells that are expressing the pattern recognition receptors, they could be macrophages, they could be dendritic cells, they could be natural killer cells, they could be epithelial cells, different cells that you already know express these pattern recognition receptors. At the same time, other components of the innate immunity, like complement, like phagocytes, they will try to contain that infection. And this is something that is going to happen very quickly, very quickly. You know, the innate immunity is a fast response. So we're talking about hours, you know, all this process is happening. And you also know that during this fast response, that innate immunity will start to produce signals in order to warn the adaptive immunity that, you know, a microbe is invading our body, and then they need to get ready to produce a specific response against that microbe. In order to do so, for example, dendritic cells, they may start to present antigens of that microbe to components of the adaptive immunity. In this case, it's showing that antigen presentation to T lymphocytes. Also, B lymphocytes may start to recognize the presence of that microbe, and then those clones of B cells that have the red receptor for that particular microbe, they will start to get activated, they will differentiate, proliferate, and at some point they will start to produce antibodies. Now, when we talk about the adaptive immunity being produced, you know, we're talking about a longer process here, especially if this is the first time that we see that particular microbe, that our immune system see that particular microbe. It could take up to 7 days. However, in order to get, you know, high levels of, for example, antibodies, it could take longer. It could take between 10, 14 days before that adaptive immunity can really do its job very well. So after this brief introduction on how the innate, you know, takes the first steps and then warns the adaptive immunity that they need to now produce a specific response. After this brief introduction, let's now take a look at the anatomy of the lymphatic system. You already know from anatomy and physiology that this is a system that has primary organs, like the case of bone marrow and the thymus, and then also has what we call secondary lymphoid organs, like lymph nodes, like tonsils, like the case of skin-associated lymphoid tissue, and mucosa-associated lymphoid tissue. The spleen is also a secondary lymphoid organ. And in the case of the bone marrow is where many of the cells of the immune system will be generated, including the T cells and the B cells. So both these cells, they will be produced in the bone marrow. They will be generated there, but while the B cells mature in the bone marrow, the T cells, they have to be taken to the thymus in order to mature. And that's where the name of T cell comes from, thymus, and B cells comes from bone marrow, the places where these cells mature. So this is just basically summarizing what I just mentioned about the primary lymphoid organs and how B cells mature in bone marrow, and T cells, they have to migrate to the thymus in order to mature there. And once these cells mature, then these naive B cells and T cells will start to circulate to hopefully find at some point, the antigen that they were made for. And in that case, they will be activated, and then now they could go through different processes of differentiation and proliferation as part of that, immune response. This slide now is showing you the anatomy of a particular secondary lymphoid organ. This is showing you what the mucosa associated lymphoid tissue looks like. In particular, the one that you find in the gastrointestinal tract, where the Paget's patches are localized under the epithelium that lines the small intestine, for example. There is a particular cell here in this epithelium that is lining the small intestine, the M cells, whose function is to sample, anything that is in the lumen of that small intestine, and to take it under that epithelium, so that now cells of the immune system can be exposed to that particular antigen, and they can start to produce an immune response against those components that may arrive into the lumen of that intestine. And this gives you an example of how this secondary lymphoid organ is structured and what is the function, in order to produce an immune response there. Something important about these M cells, you will see in future classes how some pathogens, they may exploit this M cell as a way to gain access to this subepithelium, area. Especially there is a pathogen, bacteria called Shigella, that does this very well. And we will learn in future classes how Shigella exploit these M cells to gain access to this area under the epithelium in order to start the infection. So with this, adaptive immunity, basically our body is able to build two important responses. Responses that naturally allow us to fight infections, but also they can be exploited to generate immunity through vaccination. The first one that I'm showing you here is the humoral response. And right here, what you're seeing here is the cell-mediated response. So humoral response and cell-mediated response. The humoral immunity right here, this one is the one that is related to the production of antibodies. The cellular immunity right here is the one that is going to coordinate the immune response, but also will be responsible for clearing infections by pathogens that enter our cells. The humoral and cellular immunity are I think they're summarized very well in this scheme that you're seeing right now. You have to keep in mind that we have a broad repertoire of different B and T cells circulating in our body. Each B and T cell clone has different specificities, able to respond to different antigens or to the same antigen, but with different affinities. For example, let's start with humoral response. When a naive B cell, and the name of naive B cell means this is a B cell that still has not encountered the antigen that they are made for. But when that naive cell finally encounters that antigen, that naive cell will be activated and now is going to differentiate and proliferate to produce plasma cells. Plasma cells are B cells that are now able to produce antibody, to secrete antibodies. But also in that process of proliferation and differentiation, they may differentiate into memory cells. Memory cells that now, in a future encounter, will be able to quickly switch to plasma cells to produce antibodies. Now, in the case of cellular immunity, that you see here to the right, basically, we have two main components there in cellular immunity. We have T helper cells, and these are cells that express on the surface the CD4 molecule. That's why they're also called, CD4 positive T cells. And then you have the cytotoxic T cells, and these are the ones that express on the surface of the cell the molecule CD8, and that's why they're called CD8 positive T cells. These cells, at the very beginning when they are produced in the bone marrow and then they are transferred to the thymus to mature, at this point they're going to be considered naive T cells because they have not encountered the antigen for which they're made yet. As soon as a antigen presenting cell, like this dendritic cell, presents that antigen to them, then now they could be activated. Now, in the case of T helper cells, that process of antigen presentation can happen through MHC class II molecules that are expressed only on the surface of antigen presenting cells like macrophages, dendritic cells, and then also B cells express the MHC class II. In the case of CD8 positive T cells, cytotoxic T cells, the antigen presentation to these cells will occur through a different molecule, through the MHC class I that is expressed by every cell that has a nucleus. There is one particular cell in our body that doesn't have a nucleus, and that's mature red blood cells. So mature red blood cells, they do not express MHC class 1. And you will see in future classes how some pathogens exploit these in order to hide from the immune system. In particular, malaria is a pathogen that is able to infect red blood cells, and the fact that they lack MHC class 1, the red blood cells, that help that parasite to hide inside of those cells. Now, these T helper cells, after they get exposed to the antigen for that particular clone of T cell, let's say T helper cell, they also will differentiate and proliferate to produce effector cells. Effector cells that now can produce cytokines. Cytokines that may help macrophages to become better in the process of phagocytosis. And this is a typical example of interaction of the adaptive immunity with the innate immunity, because the process of phagocytosis, you already know, is part of the innate immunity. And here you see how these T helper cells, part of the adaptive immunity, are producing cytokines, signals to make this macrophage perform phagocytosis even better. Also, these effector T helper cells that get activated, they may also produce cytokines that may aid in the process of B cells, in the process of differentiation, and in the process of proliferation of B cells. And this will be particularly important in a particular group of antigens that we call T-dependent antigens. Now, in the case of cytotoxic T cells, you know, after they receive the signal from this cell that expresses MHC class 1, it could be any cell that has a nucleus, then now they can be also activated, they can proliferate, differentiate, and become effector cells. Effector cells that now may identify abnormal cells in our body that express that particular antigen, and they could destroy those cells that are abnormal. So let's take a look now at these, well, you already know, I already mentioned to you what naive means and what activated means. You know, a naive lymphocyte is a lymphocyte that has not found the antigen that they are made for yet. An activated lymphocyte is a lymphocyte that found that antigen. That lymphocyte now is not naive anymore. It has been activated, and it could be differentiated into effector lymphocytes or memory lymphocytes. You have effector lymphocytes for T cells, T helper and T cytotoxic cells, and you also have effector lymphocytes for B cells. That would be the case of the plasma cells that secrete antibodies. And then for both T cells and B cells, you can also produce memory lymphocytes. These are a particular group of lymphocytes that, you know, are created after that encounter with that antigen. And the function of these memory lymphocytes is that if we now get exposed to that same antigen again in the future, these memory lymphocytes, they can switch very quickly to effector lymphocytes. And then now that response that could take some time after when we have the first ever encounter with the antigen. After the second, you know, encounter, what we call a secondary response, these memory cells will be able to differentiate so quickly into these effector lymphocytes that then now it's not going to require those 7 days, you know, that I mentioned before in the process of generating this adaptive immunity. That's the function of those memory lymphocytes, to guarantee that in the next encounter with that antigen, the response is going to be faster and stronger against that antigen. So we're talking about antigens, right? And let's first review some important characteristics of antigens. So basically, an antigen right here, or the abbreviation of that would be AG, is a molecule or a molecular structure that do not belong into our body. And then now the immune system recognizes it and produce a specific response against that. It could be, for example, a molecule that is maybe present at on the outside of a pathogen. And this name, antigen, stands from antibody generator. It was called like that at the very beginning because, years ago, we first thought that antibodies were the only components that were able to generate, interact and with these antigens. But now we know that there is a cellular component, cellular immunity, that also can be activated by different antigens. So antigens are not generating antibodies only. Antigens can also generate cellular immunity, can also activate these T cells that I was mentioning to you before, the CD4 positive T cells, the CD8 positive T cells. There are two main categories of antigens, and you're going to understand this soon. We're going to get into that in a minute. And it's the T-dependent antigens and the T-independent antigens. T-dependent antigens are antigens that when the B naive cell is recognized by that antigen and interacts with that B cell, this T-dependent antigen, the B cell that is responding to that antigen will require the help from T helper cells. And that's why it's called T-dependent. Now, T-independent antigens are antigens that when they bind to the surface of the B cell that will be activated by that particular antigen, those B cells will be able to get activated without the help of T helper cells. And I will tell you soon why that happens. Now, an important concept that you need to understand about antigens is that the response to antigens could vary, depending on the type of antigen that is present. We may find, for example, molecules that are too small and they may not elicit an immune response. We call those molecules that are too small to produce an immune response, we call them aptens. But if we're talking about a larger molecule, like a protein, for example, they generally elicit a strong immune response. Lipids usually are not very immunogenic. Keep that in mind. Now, something about antigen is that an antigen could be a very complex molecule that could have different parts, different structures. Each of them could produce, could bind to different antibodies, could target different clones of B cells or T cells. Now, the portions of the antigen to which, the immune response is being directed, we call those epitopes, the portion of that antigen. Usually The way that I describe this, imagine that your head is an antigen, is a molecule that is strange to our body and that is going to produce an immune response. Now, there are different parts in your head that could become a target, for antibodies or for cellular immunity. Maybe your nose, maybe your ears, maybe your teeth. Each of them with a different conformation, a particular structure, each of them could be a target. And we're going to call each of these portions of the antigen that can be recognized by the immune system, we're going to call them epitopes. Another name for the same thing is antigenic determinants. So antigenic determinants and epitope is the same. Two names, for the same thing. And you could imagine that, if you look at a whole cell, like a bacterial cell, there could be then now so many potential targets there that could become, a target for our immune system. Maybe epitopes in the pili, like it's shown here. Maybe epitopes on the flagella. Maybe epitopes on the capsule. Maybe epitopes on the cell wall. There could be many different, structures that could be recognized by our immune system. Let's now take a look at antibodies. And in this slide, I am showing you the basic structure of a monomeric antibody, right? Monomeric, made of one molecule. This is the basic structure of a monomeric antibody. A particular antibody is always produced in response to the differentiation and proliferation of B clone cells that now produce this very specific antibody that binds to the antigen that has arrived to our body. The antibodies, they bind to the antigen via the fragment antigen binding FAB region in this area. And here you see more specifically what we call the antigen binding site. Each tip of the Y shape, this is a Y shape that you have here. Each tip there, of each of these arms that you see here, that will be the site to which the antigen binds. And that is what we call, again, the antigen binding site. The best analogy here is imagine that right here, the antigen binding site is is a lock, and then the antigen is the key. They have to fit perfectly with each other in order to allow that antibody to specifically bind to that antigen. Okay, so now the ability of an antibody to communicate with other components of the immune system is usually mediated by this other region that you see here, the Fc region of that antibody. The antibodies are typically made of these basic structural units that you see here, each with two heavy chains and two light chains. There are different types of antibodies, heavy chains, that define the 5 different types of Fc fragments, Fc region, right, that we know. We have IgM, we have IgG, IgA, IgE, and finally IgD. Let's take a look at the functions that antibodies can have. This slide is summarizing that. Sorry. So a typical function of an antibody could be neutralization. Let's say a virus arrives and B cells that produce antibodies against that virus are activated, they differentiate, proliferate, they start to produce these antibodies, and now the antibody binds to the surface of the virus. What happened now? The virus is not going to be able to bind to the receptor because the antibodies are neutralizing the virus, not allowing the virus to interact with the receptor. Similarly, those antibodies could be produced against a toxin produced by a bacteria. After those antibodies bind to that toxin, they will neutralize that toxin, not allowing the toxin to reach, the final target. Another function that antibodies can have is opsonization. We discussed opsonization in the innate immunity mediated by the C3b complement protein. Well, antibodies can do similar thing. They can cover all over, a microbe like this. And then now the phagocytes, they have receptors for the Fc region of those antibodies that are coding the pathogen. And this will now facilitate grabbing that bacteria and engulfing that and taking that into the phagocyte to destroy it inside of the cell. You also know from the innate immunity class that there is a classical pathway to activate complement that is mediated by antibodies. The antibodies bind to the surface of the pathogen, and now the complement system proteins can bind and activate the complement system. The antibodies that mediate the classical pathway of the complement are usually IgM and IgG. The other classes or subtype of antibodies, they are not involved in the complement system activation. Then also imagine an antibody that targets, that bind to the flagella, and this binding now, because the flagella cannot move properly anymore, will immobilize that bacteria, not allowing that bacteria to move freely. Another function, cross-linking. Sorry. And basically, in cross-linking, what is going to happen here is the antibody, because it has two arms, can bind to two different cells at the same time. And it will create kind of like a network of antibodies with bacteria, will aggregate them, and it will make it more easy now for the immune system to get ready of that pathogen. And then the final function that antibodies can have is this antibody-dependent cellular cytotoxicity, ADCC. In this case, we're talking about antibodies that bind to the surface of a cell that maybe is infected with a virus, or maybe this is a cancerous cell that is abnormal, and those antibodies are able to recognize that abnormal cell. And after they bind to the surface of that cell, natural killer cells, they have the ability to recognize the Fc region of those antibodies that are already bound to the cell. And these natural killer cells, they will start to produce enzymes and proteins that will destroy that cell. So those are the different functions that antibodies can have. Let's now take a look at the structure of the different antibody subtypes or classes of antibodies. And I'm going to start with IgM right here. And I will start with IgM for a main reason, and it's that every B cell, every naive B cell, when they start in the process of activation and they become plasma cells, at the very beginning, they will start producing always IgM. And depending on the nature of the antigen, especially if it's a T-dependent antigen, those cells may switch and start to produce all of a sudden, different antibodies. They may switch to IgG or IgA or IgE. Now, in terms of the structure, IgM is the largest antibody that we know, and it's a pentameric antibody, and that means it's made of 5 monomers so I'm showing you here 5 monomers of antibodies that now get connected and this pentameric IgM is usually, the main functions of that IgM could be, well, activation of the classical pathway. You already know that for the complement. Could participate in opsonization. And cytolysis. And this IgM usually it circulates systemically in the bloodstream, but it could be also present in some mucosal surfaces. Like for example, the gut lumen. It could be also present in the breast milk. So those are the main things that I want you to know about IgM. Let's now talk right now about IgA right here. IgA is an antibody that can be monomeric, just one molecule, or dimeric, 2 of them. The IgA dimeric form is the most prevalent IgA, and is also called the secretory IgA. The acronym would be this. And this is the main immunoglobulin that can be found in mucus secretions. You can find these immunoglobulin, for example, in tears, saliva, sweat, colostrum, and also in secretions of the genitourinary tract, gastrointestinal tract, prostate, respiratory epithelium. You're going to find a lot of these secretory IgA. The main functions of the IgA is neutralization, cross-linking, and immobilization. Those are the main functions for the IgA. The next antibody, oh, by the way, something that you will hear, from, I will show you when we talk about pathogens and how they evade the immune system. You will see how some pathogens, they can produce enzymes that will destroy that IgA. And that is the case of, for example, gonorrhea. The bacteria that produces gonorrhea is able to produce enzymes that target the IgA. This is the scientific name of the bacteria that produces gonorrhea. And you will see in future classes, the different mechanisms that this bacteria has to evade the immune system, including the ability to produce proteases, enzymes that can destroy the IgA that we produce. Let's now talk about IgE right here. IgE is a basic monomeric antibody. And the main function of IgE is immunity against parasites. That is the main function of IgE. It's also associated to hypersensitivity, type 1, that is basically allergy, right? We will talk about that soon in a class. And I think that's what I want you to know right now about the IgE. And then finally, IgG. Here we're talking about the most common antibody circulating in the bloodstream. Represents about 75% of serum antibodies. The most common one. It's also a monomeric antibody. And there are 4 different subclasses of IgG. So you have IgG1, IgG2, IgG3, and IgG4. These antibodies, the antibody that probably is the more versatile in terms of the functions that can perform. And together with IgM is the antibody that can activate the classical pathway for the complement activation. It's also the only antibody that can cross the placenta. And the next slide is giving you more information about the importance of this IgG that is able to cross the placenta. Basically, what this graph is showing you here is that the levels of IgG in a fetus, right here before birth, and then after birth, what are the levels of those IgG and where are those IgG coming from after birth in that infant. So basically, while that fetus is in the inside the mother, it's going to rely on the mother antibodies that are this light blue color that you see here. The dark blue is the total, dark blue is total antibodies. During this period, those total antibodies will come only from the mother, because the fetus still doesn't have a mature immune system to produce his or her own antibodies. Now, those antibodies that are being given by the mother, that are crossing the placenta, or that also are being given to the infant through breastfeeding, those antibodies, they don't last forever. At some point, they will disappear, those maternal antibodies. And then now the infant own immune system will have to start to produce those antibodies that you see here. And at some point, they will be those antibodies produced by his or her own immune system will be the ones responsible for protecting them. But before birth and early on during birth, that infant that doesn't have a mature immune system will rely on these antibodies that are passively passed from the mother to the newborn. So now let's go into the B cell response. So we're going to be talking about humoral immunity, the production of antibodies. And we're going to start with T-dependent antigens. Remember that I mentioned to you T-dependent antigens are antigens that will activate that particular B cell clone that recognize that antigen. But then now for that B cell clone with that T-dependent antigen, in order to be activated and proliferate and differentiate, T cells, T helper cells, need to provide signals in order for that to happen. So how is this whole thing happening here? So let's say this is a naive B cell, have not been activated yet in this first step. And then all of a sudden finds the antigen floating outside in the extracellular medium. The antigen binds to the B cell receptor that is specific for that antigen. And that's why this clone, is responding to that particular antigen. That antigen will be taken into the cell, will be processed inside of the cell. And then now pieces of that antigen will be presented in the context of MHC class II molecules that B cells can produce. Different MHC class II molecules here, presenting each of them different pieces of that antigen. And now, right here, that B cell finds a T cell, a CD4 positive T cell, or T helper cell. An effector cell that brings the particular T cell receptor that is able to interact with this antigen here that is being presented in the context of MHC class II. And it's going to interact with the T cell receptor on the surface of the T helper cell, the CD4 positive T cell. If that interaction is proper and they correspond to each other, then now this T helper cell will start to produce cytokines that will allow this B cell now to get activated to proliferate and differentiate into the effector cells. Effector cells that will be the plasma cells, but also some of them will differentiate into the memory cells. Now, if by any chance the antigen that this B cell is presented in the context of MHC class II is not able to be recognized by a particular T cell receptor on the surface of the T helper cell. And now this B cell is not going to receive the signal of the cytokines required for that B cell to proliferate, differentiate. This B cell will be basically eliminated through a process that we call anergy. It will become anergic and will be destroyed. Now, I mentioned to you that sometimes there are antigens that we call T-independent antigens that they do not require the help of the T cells, the T helper cells, the CD4 positive T cells. And we're going to call these antigens T-independent antigens. These T-independent antigens, they elicit antibody production by B lymphocytes without that T lymphocyte involvement. The T-independent antigens, they have a highly repetitive structures in which maybe there is a maybe is a polymer in which a monomer is repeated over and over and over and over. That would be the case, for example, of polysaccharides. Another T-independent antigen could be lipopolysaccharides. And sometimes you may also find polymeric proteins that also have this repetitive structure. Now, when you have that repetitive structure, that causes several B cell receptors on the surface of the naive B cell, they could recognize several of those receptors at the same time, the antigen. And when you have several receptors binding to the same molecule of that antigen, this will produce what we call cross-linking. And this will become the signal that will activate that B cell. And now that B cell can differentiate and proliferate, differentiate and proliferate. Now, when you have T-independent antigens, generally this B cell will be able to produce only IgM because it's not receiving the signal from a T helper cell, the cytokines that will mediate the switching of class to another type class of antibody. So this one cannot switch from IgM with these particular antigens. This B cell will not switch and will be just producing IgM. And these are usually, low affinity IgM. Usually T-independent antigens, they don't produce a strong immune response. And usually it's a response that doesn't last for too long. This other slide here is, showing you also related to the human immunity here, characteristic of the primary response versus the secondary response. What this is telling you is that when you first encounter a particular antigen, when your immune system sees for the first time a particular antigen right here, that first encounter will result in a response that takes some time. Remember the 7 days and then about 10-14 days to reach good levels of that antibody here. Now, if your immune system encounter the same antigen in the future, because there are now memory cells circulating from the first encounter, the next encounter will result in a faster and stronger response. And that's the main difference between secondary response and primary response. The secondary response is faster and stronger. Now, I'll also look here that, you know, when the antigen presentation happens, at the very beginning, it would be IgM. And this looks like this is a T-dependent antigen that is allowing now those B cells to switch to IgG. And usually IgM, you know, doesn't last for too long. And every time that that encounter happens again at the very beginning, because there are some naive cells that are being activated also during the second encounter, some IgM is going to be produced, but then they will switch quickly to the production of IgG in this particular case. It looks like a T-dependent antigen. And this is, you know, giving you more information about the characteristics of the secondary response, what I already showed you in the previous picture. Basically, that when we're talking about a secondary response, it's going to be faster, it's going to be stronger. And the reason for this is that there are now memory B cells from the primary response that are circulating that will be responsible for that greater number of cells that can be quickly activated, that can proliferate, differentiate, and start to produce more of that antibody. So in few words, secondary response is faster and stronger than the primary response.